Okay. Okay. Allele. Remember allele. Version of a gene. We say the allele you got from your mom, or the allele you got from your dad. Okay. Which of the patterns can't belong to a child of this couple? Okay, so here's mom, and here's dad. Okay, so A could be, and so this one could be a kid, absolutely. This is dad's allele, and this is mom's allele, so that's good. This is mom's allele, and this is also mom's allele, so this can't be it. Okay? And then this can be mom and dad. Mom and dad. Okay? Which cannot be the child of this couple? Huh? C? Now remember one thing here. This right here is an example of a double band. Okay? So, in other words, mom has it and dad has it. So that could definitely be both. Okay? So that's okay. A is, this is dad, and this is mom. And then this is dad, and this is mom. And then this is a double band too, but mom doesn't have one, so this cannot be it. Okay. What kind of cell is this? Right. Okay. Why do we know that? What do we have here? We have a nucleus. Okay? Bacteria or archaea do not have a nucleus, nor do they have these things called mitochondria or any of the other stuff. They just have DNA inside of a cell, no nucleus or anything like that. So only a eukaryotic cell has all this other stuff. So who are these guys? A, B, or C? Oh, that's not right. Yeah, okay. B, C. Okay, now what? B. Okay. Yep, Watson and Crick. Sorry about that, I'll have to fix that. And the reason they figured it out actually was because they took, you guys haven't ever taken chemistry, maybe in high school. Well, sometimes you get these different atoms and you actually put them together into a molecule. Well, they pretty much did it that way. And it helped them figure out where things had to go. Okay. So, what goes here? What goes here? Okay, good. Okay, so what we have is we have a double-stranded helix, remember? So, double-stranded, twisted. When there's replication of the cell, you have to replicate all the DNA. So what you do is, all this DNA unwinds, and it then adds the complementary strand with something called DNA polymerase. An investigator finds a small amount of tissue at a crime scene. What procedure will they use to make the small amount into a large amount? PCR, not STR. Okay. STRs are highly repetitive variable. In other words, they're variable, and there's a lot of repeats in them. And they vary among people. Okay, so short tandem repeats. They use about, 16 or 17 or more now. Almost no other person will share the same length of the repeats at each of those specific sites. And then to actually separate the short tandem repeats, whether they're 2 repeats or 5 repeats or 7 repeats, you use something called gel electrophoresis. You actually put the DNA in these different wells, and then you run it, and you can see, remember something, larger fragments move more slowly, so they stay at the top, and smaller fragments move more quickly, so they're down at the bottom. And here's the genetic code. It turns out that when you see, let's just take one little look at it. Okay. I mentioned, like right here, that's TTG, right? Remember, it takes 3 base pairs to make an amino acid. And these are the different amino acids. You don't have to know them all. But this one, TTC, makes leucine. Okay? Some of these are redundant, but in general, you've got 20 amino acids. If you remember back from the molecules we were studying, 20 amino acids. Some of them we can make ourselves, and some of them are called essential amino acids. Those are about 7 of them. Okay, and then we'll stop here, and we'll start again on Monday.